Hello, I'm AdamSigwing. This is my seminar presentation exploring positive versus negative feedback mechanisms in maintaining prairie biodiversity. So grasslands are highly diverse ecosystems with robust communities that have evolved to survive harsh conditions. Grasslands are high in biodiversity as there's an average of 40 to 60 grass species, 300 plus wildflower species, and over 100 different woody and shrub species. But with such extreme environmental factors and high density of individuals in a given area, how are there so many different species able to coexist and even thrive to be one of the most productive ecosystems? One model that can help explain coexistence in highly diverse areas is resource partitioning. Plants and grasslands have evolved and adapted to survive harsh conditions by specialization, therefore filling different niches in an ecosystem. Prairie species are known for their massive root systems that can easily surpass the above-ground height of the plant. Resource partitioning can occur in the root zone with water absorption, for example. Grasses, forbs, and shrubs make up a large portion of plants in the grasslands, but they focus their roots at different levels in the soil, as shown in the figures below. Grasses will absorb the majority of the water they need in the top 6 inches of the soil. Forbs and wildflowers will absorb from the same depth and compete with the grasses, but they'll focus more of their energy in the roots below the dense root mass of the grasses. Shrubs will focus their energy more so on their deeper root systems to reach moisture that neither grasses nor forbs can. Due to these specializations, these plants can be found in close proximity to each other and not directly compete for water, allowing for unimpeded growth. An alternative model to plant coexistence is feedback loops with soil pathogens. The rhizosphere hosts most of the mutualistic relationships between plant and soil microbes. Soil pathogens can also inhabit the zone and have co-evolved with plants, but in a parasitic manner. Both the plants and pathogens have evolved to combat each other's defenses, which has led to host-specific pathogens, which are pathogens that can only attack a certain plant species. This can have severe negative effects on monocultures such as land used for agriculture, but it can also help maintain a healthy and diverse ecosystem. Plants can prevent one species from becoming overly dominant by reducing that species' population, which will allow other species to flourish in that area. This concept leads to the negative feedback loop, which is when forces are counterbalancing each other to create an equilibrium, as shown in the figure in the bottom right. This study is part of the National Science Foundation's Dimensions of Biodiversity program that is being led by the researchers at KU. This is a multi-site study that included several colleges and universities that are located within the tallgrass prairie bioregion, but in different precipitation zones. These sites include Eastern Illinois University, Illinois State University, Baker University, University of Kansas, Johnson County Community College, Bethel College, and McPherson College. Each site followed the same guidelines and procedures as well as to communicate issues, sharing data and information with each other. The experiment can be split into 2 different phases. The first phase included establishing a field site to inoculate soil. The second phase will use that inoculated soil in a greenhouse feedback experiment to determine which feedback mechanisms are in play. Here's just a map showing the precipitation gradient of the sites. In the east, we have the Illinois State and Eastern Illinois University, which receives anywhere from 800 to 1,000 milliliters of precipitation a year. In Northeast Kansas, we have Baker, KU, Johnson County Community College, which receives mid-700s to closer to 900 milliliters of precipitation a year. Whereas in Central Kansas with us and McPherson College, we just receive around 600 to mid-700s. In this study, we are using 3 native prairie species that each represent a different group within the prairie ecosystem. The Lespedeza capita represents the legume family, which has great nitrogen fixation properties and extremely drought tolerant. The Silphium is part of the sunflower family, which of course has great pollinators, especially bees that are attracted to it. The Andropogon, also known as big blue stem, as many of us are familiar with it, is one of the 4 major grasses found within tallgrass prairies. So there's 2 main outcomes that could come out from this experiment. If negative feedback loops are operating, we should see a decreased plant growth with an increase in frequency of specific soil microbiota. This can be kind of shown in the figure in the bottom left. If positive feedback loops are operating, we should see an increased plant growth with an increase of specific soil microbiota, which is seen in the lower right figure. So to start the field site, we first needed to collect 12 gallons of soil from a virgin prairie, which is a prairie that has not been majorly disturbed or turned into farmland, and then also soil from a restored prairie site. The virgin prairie that was located the virgin prairie that was used was located at Chase State Fishing Lake, which was largely been left undisturbed by humans. And then the restored prairie was the prairie east of Bethel College, which was established in 2007 on former agricultural land. The reason behind sampling from two sites is to accurately represent soil microbes. The microbial composition of a restored prairie is vastly different from a virgin prairie. So using both, we should have sufficient richness of microbes that are often associated with the three plant species. Smaller samples were then taken from this and sent to KU for analysis over the microbial community. The rest was used in seedling propagation to condition the soils, or condition the plants to the soil. To get the plants ready for the field site, we first had to germinate the seeds of the three plant species so they could be transplanted later on. Initially, we sowed the seeds in the standard planting trays with potting mix. We stratified the seeds at 4 degrees Celsius in a fridge to simulate the winter season. This stratification is needed for several plants to mimic the winter, obviously, to trigger growth in the spring, because without it, they would hardly have any germination. After roughly a month and a half of cold stratification, we placed all trays in the greenhouse where they were regularly watered and cared for. Once these seedlings developed enough, they were transplanted in the groove tubes containing a mixture of prairie and field soil. Again, process can be seen on the right. The native prairie soil was included to provide native microbes as an inoculant to prepare the plants to be transplanted. To establish the field site to inoculate the soil, a 32 by 16 area was carved out of the restored prairie by Bethel's campus. Once we found a suitable location for the field site, we followed the diagram that KU provided to stake out the borders of the site as shown in the bottom figure. We then placed a thick tarp over the vegetation to prevent any sunlight or water from reaching the plants in a process called solarization. This made clearing the field site of unwanted vegetation much easier without the use of herbicides. Now the diagram on the top right shows the orientation within a plot in the field site. This layout was followed for all 18 plots. And overall, the field site was block randomized, meaning that within each column, one of each species was present and was randomly oriented. This ensures that each species was subjected to equal environmental factors. Once the unwanted vegetation has been removed, we began flagging the site according to the diagram on the previous slide. Each species was replicated 6 times, totaling 18 plots. Each plot contains 9 individual plants of one species, coming to a total of 54 individuals per species. Once every plot was flagged, we began transplanting individual plants to their corresponding spots. Once all plants were transplanted, the field site was maintained throughout the rest of the experiment. This included regular mowing to suppress plant growth in between plots and thoroughly weeding the plots themselves. This was done to ensure that we only had the target species growing in each plot. The biggest culprits were foxtail and other weeds which just grew out of control in and around the plots. To get a measure of the productivity of the plants in the field site, height and biomass measurements were taken. During the first year, biomass measurements were measured by clipping and collecting above ground biomass from all the plants in the field site and air drying them in the greenhouse to remove any moisture. This drying process gives a more accurate mass measurement. And then for the second year is when we measured the height of all the plants. Based off the recorded data and field observations, the majority of the plants did not experience any difficulties growing aside from a few outliers, which did not greatly affect the overall averages. The final portion of the project is the greenhouse experiment. This will use a series of tubes that will house one individual plant that will be replicated with different soil inoculums that were taken from each field site or each plot within the field site. Each tube will be filled with a combination of sterile soil and inoculum soil. Now this experiment is also block randomized, similar to the field site, with each block here containing 24 individuals divided into 4 groups that will receive different soil inoculum treatment. The figure in the bottom left shows an overview of how this experiment will be carried out with each block. Soil from each plot at the field site is used to expose each plant to a different group of soil microbes, while the sterile control will not control any soil inoculum. This portion is still ongoing and the data will be collected sometime midsummer. Now, COVID-19 has greatly impacted this project. The original plan was to perform the greenhouse experiment and collect data in the spring of 2020 and then partake in an REU in the summer of 2020 to analyze and present the data at a research symposium at KU. But the pandemic essentially pushed everything back a year, and since this is my senior year, I will not be able to see this project to the end. So Mia Loganville, a junior biology major who has been assisting me with this project for over a year, will complete it. She will finish the greenhouse experiment and collect data such as height and biomass that will be pooled with the other institutions that analyze during an REU this summer at KU. Then they will determine whether or not a positive or negative feedback loops are operating based on plant growth during that experiment. I would like to thank John Piper, my advisor, who has helped me navigate this project and assist with every step of it. Dwight Crable, who along with John Piper asked the REACH committee for research funds to help me carry out this project. I'd also like to thank Jim Beaver and Tara Lubin, who are the KU researchers who have led this project and organized group meetings to help everyone stay up to date and organized. I'd also like to thank the participating institutions for helping with this project and always being in communication with each other, as well as Mia Loganville, Beth Piper, Laura Tran, Sandy Dow, and Natalie Graber for their assistance in various steps throughout the project. Thank you.